Our story is about a new protein quality control mechanism that is located in a specific part of the cell, that is the nuclear membrane. So proteins are like the building blocks of a cell. They have a very important structural and functional role in the cell. But when they are synthesized just as a linear string, and they need to fold and properly to have the right structure, so then they can interact with each other and communicate with other protein in the cell. This process is not perfect, so a lot of protein, because of mutation or even just because the cell is under stress, they don't fold properly. And if they have the wrong structure, they cannot interact with each other anymore, and they can be dangerous for this reason, or they can even aggregate and cause stress and damage to the cell.
What we looked at was a pathway that gets these proteins and recognizes the proteins and degrades them, just gets rid of them, because if they are not working correctly and they are not functional, they can be bad for the cell, they can be toxic. Although some pathways had been already described by the time we found this pathway was very interesting because it was localized in a very specific part of the cell. It's in the inner nuclear membrane, which is the membrane that is surrounding the nucleus. There is a link with protein degradation and misfolded proteins with neurodegenerative diseases.
I'm not going to say that our research will provide the cure for this disease at all, but it might give some information that might be useful for characterizing this kind of protein defect and the clearance from the cell. At the first results we had, our boss started to present it into meetings, to get feedback from people and know that you are on the right track. We were not the only people working on it. Other people were looking at something similar, so we were like, okay, if we want to be the one to deliver this story, we need to do it fast.
The way the CRG manages PhDs is very helpful, and it really lets you progress scientifically and gets you where you want to be. It really provides a lot of opportunities to do whatever you want and help mostly. And yeah, we have a very strong PhD community, and we have also the graduate committee that is there for us. We are very lucky to be here, and that you notice from day one, and every day you feel, at least me, I feel very lucky to be here.